Hello everyone, I'm going to talk about DNA replication today. First, I'm going to talk about the structure. Nucleotides is composed by 5 carbon sugar, which we call deoxyribose, phosphate groups, and nitrogenous base. As you can see in the photo, nitrogenous base is composed of A, adenine, T, thymine, G, guanine, and C, cytosine. The lines between are the hydrogen bonds that connects the nitrogenous base. Between A and T, there's only 2 bonds. However, in G and C, there are 3 hydrogen bonds. The whole thing, we call it complementary pairing because the pairing between AG and TC is formed equal-sized pairing of nitrogen base. The 2 sides of the structure, we call it phosphate sugar backbone because it's formed of phosphate groups and deoxyribose sugar. Lastly, double helix is just the structure of the DNA. Next, I'm going to talk about action helicase. As you can see in the picture, action helicase is basically that the DNA double helix is unzipped into 2 strands for the replication later on. Next, RNA primase. It adds NTP to single-stranded DNA in a 5-3 directions. RNA primer is RNA polymerase added several strains of RNA to lagging strands. Next, for RNA polymerase 3, it transcribes DNA to synthesize RNA. There are 3 stages in this progress. First, initiation. It is the construction of RNA polymers. Second, elongation. It is the synthesis of RNA transcript. Third, termination. It finishes up RNA transcription and disassembly of RNA polymers. Next, I'm going to talk about lagging strands and leading strands. So for a lagging strand, the first step is to the helicase breaks down the double helix apart. And then second, the RNA primase anchors itself to DNA. Third, DNA polymerase 3 assembles nucleotides. So what is DNA polymerase 3? It adds DNTP in 5-3 direction, where RNA primase has added a short length of complementary. Last step for the leading strand would be DNA polymerase 1 remove. It removes RNA and replaces them with DNA, so it continues to the next stage, which is the lagging strand. First stage of the lagging strand is the helicase, which it separates hydrogen bonds to unwind the DNA. Second, the RNA primase, which have been mentioned before already. And then the third one, the DNA polymerase 3 make Okazaki fragments. Okazaki fragment is basically a short fragment of DNA on only the lagging strands. And then the DNA, after the DNA polymerase 3 produce Okazaki fragments, DNA polymerase 1 will remove the RNA primerase. And then the DNA ligase. Ligase would usually to glue up things, so the DNA ligase would glue Okazaki fragments together, and then to make sugar phosphate bonds, and then so the new DNA can be created. So that's basically it. Thank you.